happy to welcome you guys here. Thank you all for coming. So we'll take a look in just a second, and this afternoon Adi is going to dig into a little more details and make sure that you know GitHub and Slack and other ways of connecting. This started 2 years ago, and I think it's the thought of David Anderson, or sorry, 1 year ago, sorry, last year, and myself and Daniel Wagenaar, and in particular Adi, who did most of the hard work, are the course organizers, and we've been aided by Helen and Mary. So right off the bat, thanks to all those people, especially Helen, Mary, and Adi, for actually doing a lot of the work in getting this organized. And thanks to you guys for coming. So it used to be at the Chen's residence. We tried to have it at the Chen's residence this summer, but it was occupied. And so we now have it in the Chen's building room, this room right here. The Chen Neuroscience Institute, as you probably also know, consists of several centers. If you go to the web page, it lists all the research centers. So this is, Richard Anderson's. This is directed by Richard Anderson. This is directed by Colin Kammerer. This used to be Doris Tsao. This is the Caltech Brain Imaging Center, and so forth. And the particular one that's responsible for this is the Data Science and AI one. And if you go to this website and others like it, it has a lot of associated information. Here's a history of Caltech Neuroscience. That's pretty worth looking at for those of you that don't know Caltech Neuroscience. This is my former PhD mentor, Mark Kunishi, and other famous people, Seymour Benzer, and others that are listed on here. But in addition to these dead people, there are many living great neuroscientists in this very building and others on campus, and some of those are going to be lecturing to you. And even those who are not may be around, and so feel free to obviously interact with everybody who's here. I myself, I think I mentioned this to you on Slack. I'm here for the 2 weeks of the summer course. I'm not actually going to be in the room because I have lots of other stuff going on concurrently. But my office is right upstairs, 345 Chen, and you can reach me by email. And happy to hang out and chat. I will try to make as many of the lectures and the social events as possible. But if you want to just catch me, or if you have problems, in addition to emailing Mary and Helen, email me or find me up on the 3rd floor of this building. And so if I'm sure you've taken a look at the agenda. So it has the 10 days here, the weekend you have off, and lists the topics, the speakers for what we have in store for you. And for each of these, the schedule is going to be pretty much the same, which is that you have breakfast in the morning. We have an introductory lecture that I'm about to start. That's kind of the only sort of lecture style content for each day. There's a break, and then there's a Q&A session. And I think at least for today we'll just have it in here. But if that's not the right ambiance, we could decide to have it outside or any place else really that's available. But the default would be to have that here. And for this part, you guys need to provide questions, thoughts, etc. And the idea is to interact with whoever the speaker was for the intro lecture and kind of broaden it out into a broader Q&A. Most of the topics, the intro lectures are going to be sort of usually, today is a little bit different because it's a broad overview, but in general, they're going to be, standard, and often relatively dense lectures. And so digging into that a little bit and asking questions and having the opportunity for more detailed discussion is what we have in this slot here. And then lunch, and then the afternoon is a methods lecture and hands-on session. And then today, we have a social that's going to be right outside here. So for today, I'm going to be doing this. You're going to be thinking about what questions, mostly sort of philosophical or broader ethical questions to think about. There's a lot of them that we can talk about from 11 to 12. And then Adi is going to take over for the afternoon. Anything you want to add to this, the broad schedule here? OK, so let's so here's the schedule again, and let's dive into what we have. So what I thought I would do in this sort of intro lecture is to preface what to think about when doing data analysis, in addition to doing data analysis, which is all the broader context. And of course, it depends a lot on the type of data that you're looking at. In my lab, we study humans, and so we deal a lot with human data. And there's a lot of ethical, IRB-related issues that may not be applicable if you're using data, from a rat or from a fly or some other organism. But some of these broader questions are in the realm of philosophy of science, and just generally about how to think about doing data analysis and how to do it responsibly. So here they are. And I will spend just a few minutes on the first one, and then these other ones will come up as we go through a few examples that I'll go over with you. But one big one, which philosophers of science, of course, have thought a lot about, and probably the only speaker who will mention it to you is Friedrich Eberhard, whose lecture is next week, which is if you're analyzing data, you have some objective, some goal in mind. And of course, this could vary hugely. I mean, it could be very simple from just, I want to make my boss happy so that I get a pay raise at the company I'm working at, or something like that, right? Or it could be something less silly than that. That is, you're just looking at some way to predict something from the data. But typically, as scientists, you want something that amounts to an explanation, either of how sensory stimuli cause or somehow related to the neural data that you're analyzing, something about the dynamics or a way to understand and reduce the dimensionality of the data itself, or a way in which to understand how the data and the stimuli through the data link to behavior, which is the typical route. But throughout all of that, you've heard all of these terms, and they're well studied and well debated within philosophy, but it's worth thinking about them. We'll think about them in the next 5 minutes, just quickly. What would count as an explanation, and what do you mean by these terms? Typically, what you're looking at are associations, correlations of some kind, from which you want to infer some kind of a model of what's going on that makes stronger claims, in particular claims about constitution or causality. But it's not obvious what these mean, and it's certainly not obvious, or it's not defined, unless you put some thought into it, typically ahead of time, to ask yourself, what's the question? What am I trying to explain with my neural data? Are these actually the appropriate data to look at my question? And so these are meta questions that if you're in the weeds and you're analyzing data, you might think are silly or in the realm of philosophy, but they're extremely important, and they're ones that you would want to think about, I think, throughout data analysis, but certainly when you begin data analysis, because otherwise you might go down a long road and analyze a lot of data and come up with something that's just not actually what it is that you were interested in explaining, right? Generalization is a huge problem, and we face it in our work all the time. But you typically want to generalize beyond the samples that you have available to say these are samples from a population of data, or you resample your data, or you use cross-validation or something to at least check to what extent the data generalize, at least within some larger corpus of data. But often you want to generalize over multiple animals, labs, or even species, right? In the ideal case, you might want to come up with something about, this is a line attractor describes fear across mammalian species, in the hypothalamus, or something broad. Of course, that's hard, but one question is, to what extent are you interested in that, and to what extent are you going to put this in the abstract, the title of your paper? Validity is related and also a very difficult question. Typically, all you have are associations between variables, but often what you're interested in is something that you can't measure directly, that you need to infer, that's a latent variable. So you might be manipulating stimuli, like cat urine odor, or an overhead shadow of a hawk, or something that induces a certain behavior in a mouse, and the mouse freezes or runs away. And so you have various neural data, you have various stimuli, you have the behaviors, but typically somewhere in there you would infer in this particular example, a latent internal state, say, of fear. That you induce fear in a mouse, you're looking at the neural correlates of that, and you're measuring fear in terms of the mouse freezing. But is it? Well, David Anderson and I wrote a book about this, and it continues to be debated. How would you know, right? And if you talk to many people on the street or psychologists, they would say, no, you can't use the word fear because you don't know what it is that the rat or the mouse is actually feeling. How do you know? Maybe it's anger. Maybe it's nothing. Depends on what how you define that. So validity is very difficult to define, but it's implicit and in a lot of what you analyze in the extent to which you think this generalizes. And then the last one is sort of more practical and becoming more and more so with the availability of shared data. And again, it goes for all data, but human subjects data is sort of the most obvious. But if you analyze somebody else's data set, or you're looking at some data, or you're sharing data, then you have responsibility for where it comes from, how other people are going to use it, what other people might do with it, what the press might do with it, et cetera. So there's broader ethical issues that amount to doing science as a responsible scientist. So these are the sort of, we're not going to talk about all of these in the next 45 minutes in any detail, and they're not even things that are very good, I think, to itemize. They amount to just saying, look, step back and think before you dive into the data, and do so throughout. So, what we ultimately, one example of what many of us ultimately want to understand is something like this, which you've probably seen before from Misha Arens. This is light sheet imaging of a larval zebrafish that shows, speeded up, the activity, or rather calcium imaging that is supposed to be an indicator of the neural activity of all the 100,000 or so neurons in this animal. So it's some complex pattern in space and time that you see in terms of all the neurons firing away here. And the challenge for us as neuroscientists is to understand how stimuli cause this, understand how all these neurons have causal relations with one another, and ultimately, if this zebrafish weren't embedded in a gel, how this would give rise to behavior, and the animal would swim around, avoid, and seek out certain goals, et cetera, and how you would scale this up to the 80 billion or so neurons in a human brain. But so if you think about that very broadly, and this slide is from work that I did with Frederick Eberhardt some time ago, here's one way of thinking about it, which gets to these terms of constitution and causality that I alluded to. So you have a brain, and you typically have latent variables that are in the domain of psychology, but I would actually argue that most of you, although you might not know it, are doing psychology, or what the future of psychology should be, which is there would be variables that may have very different names in the future psychology about memory, attention, or things like personality intelligence. And these are in the brain in the sense that they would be constituted by the brain. So the way that scientifically you should think about that, the way you would think about memory or attention, personality intelligence, is that these are actually macro variables constituted by presumably some very complex pattern of activity in the neurons of a brain, as opposed to caused by the brain, in which case you would be a dualist and you would drop out of this room. So they're constituted by that, and you cannot manipulate them independently, which is the criterion for constitution as opposed to causality. So I can't change your personality without doing something in your brain. They're not independent, not independently manipulable. These then have causal relations with what we measure. And so in the simplest case, what you measure is something of behavior. So you could, in the human case here that's illustrated, have personality and intelligence with various tests or questionnaires. And you could measure this in the brain, which is in the experiment, in the paper that I'll go through as one example next, what was done there. So this is a correlation, a triangle symmetric correlation matrix between all the pairwise regions in the brain that were sampled here. And so this is also caused in a very indirect and complicated and poorly understood way by activity of neurons. It causes changes in blood flow, causes changes in local magnetic susceptibility that we can measure with bold fMRI. So you have two sets of dependent measures, behavioral measures, neural measures, and in many cases, like the example that I'll show in a minute, you want to relate those two. And ultimately, what you would like is some kind of a mechanism that is from the neural data, which are observational, not interventional in this example, you would infer a mechanism. This brain, this neuron has a causal effect on this neuron and that neuron. It's going to be extremely complicated, like the light sheet imaging of the zebrafish, only much more so. That explains the person's personality, intelligence, and their performance on these various tasks. So that's the rough picture. But again, the thought, let me just skip this one here. The crucial thing to keep in mind is the distinction between causal and constitutive relations in what you want to explain and whether or not the data are suitable for that. This picture is actually taken from some reference that I didn't cite, which is probably just as well, because it's a bad picture in the sense that it conflates these two and doesn't make clear which is constitution and causation. But you often see this, I mean, these are just dependencies here, but you often see pictures like this in sort of mathematical psychology papers. In this instance, they're schematizing how to think about general intelligence. They think there's some neural basis. You would now, given what I just told you, say that these arrows are constitutive rather than causal relations. So the neural basis is the general intelligence. That's just what it is. But the general intelligence causes the behavior. How can you tell? You can manipulate them independently, right? I can give you curare and make you completely paralyzed. The dependent measures are all zero. Have I changed your intelligence? No. So clearly the intelligence is separate. That is, it causes the behavioral measures. Whereas in the brain, if I do something to your brain to actually decrease your intelligence, I can't manipulate those independently. I would change both, right? I'm not going to say much about this. The picture is way too complicated, but I can, if you close your eyes and listen to what I'm saying, it's probably easier than trying to decipher the images. But later on, Iman and Frederick next week in their lecture are going to look at this and actually walk you through some of the code that Iman has for this. One big question throughout all of this is, well, what are the data, the type, or at what level of grain or aggregation are the data most useful to go through this kind of exercise, to have causal and constitutive relations, to have some kind of explanation. And it's not obvious how to do that. So if you go to the neural example, you might say, well, look, this is useless right off the start. Bold fMRI is very macroscopic. What we want are single neurons and action potentials. Or you might be even more extreme, and you might want to say, I want to measure potential changes throughout the entire dendritic tree of a neuron. It's already really complicated. Or you might say, no, look, we need to look at population activity in neurons and circuits, or something even broader than that, which is what fMRI looks at, and large macroscopic networks. Well, it's not clear. You can do dimensionality reduction on all the 80 billion, the activity of all the 80 billion neurons or so in the human brain by itself, but that's not really what you want in this case. What you want is some way of dimensionality reduction or some kind of aggregation of the micro data to more macroscopic variables such that these actually explain what it is that you're interested in explaining, which depends on how you aggregate these, right? So in the mouse example, you're recording from lots of neurons, and you have lots of different ways of quantifying the behavior. But in both of those cases, you want to somehow aggregate the neurons and look at some population level phenomenon. And on the behavior, you want to say the mouse is freezing or the mouse is running away, not the left paw moved by 1 millimeter to the left. That's not the level at which this is meaningful. And same thing here. So in the human case, typically you want to aggregate responses on questionnaires. You might want to derive a factor from an exploratory factor analysis and say there's some network activity in the human brain measured by fMRI that's related to neuroticism or extroversion or whatever the personality dimension is, okay? But so these are, again, maybe these are obvious to you, but they're super important and they're difficult questions, which is why you would want to think about them before you start diving deep into data analysis. So 2 quick examples here from studies in my own lab. The first one neural, the second not even neural, but many of the same points apply. So some time ago, Julien Dubois, who's now something like chief scientific officer at Kernel, decided to look at an existing database, the Human Connectome Project, which has about 1,000 subjects, and ask the question, can we, from the resting state brain activity of the subjects in that database, predict anything about their personality? And so we had both of these measures available. We have the personality of these 1,000 subjects. So first thing we do is we take a look at their distribution. Does it make sense? How does it look? And it makes sense. These people are mostly open, conscientious, extrovert, agreeable, and not very neurotic is what this shows, right? So that's one set of data, the behavioral data. For the same subjects, we then have the neural data, which were these resting state functional connectivity matrices of fMRI data. Again, the details here really don't matter. You have a set of neural data, a set of behavioral data in 1,000 subjects. In this particular example, the goal was to get to this title and be able to predict personality from resting state connectivity data. So before doing any of this, and in fact we were forced to think about this because you can't just get these data. You need to send in an application to the Human Connectome Project, to NIH, to get access to the data. You need to explain a little bit about what you want to use these data for. You don't get access to all of the data because some of them are more risky or have a higher probability of identifiability of the subjects, et cetera, et cetera. So there's a whole bunch of preliminary work. We have to put in a proposal to the Institutional Review Board here at Caltech. So a lot of planning and study inception. And again, if you're just playing around with a data set without any particular goal, as you're going to be doing in this course, of course that's fine. But if you're aiming for a thesis or a paper, anything you're going to publish, an actual project, you want to start thinking about all of these. And it depends on the type of data. Human subjects data probably has more consideration, certainly with respect to ethics, than data from non-human animals. But pretty much all of this is going to apply to some extent. And there are well-known resources. For instance, Open Science Framework, we use pretty much all the time now, and people doing human neuroscience studies, fMRI, many people use this. Maybe not most, but many. It's not that common for non-human neural data, but it's still a good idea, even if you do it informally, to have some plan. If you do it full-fledged on the Open Science Framework, you actually literally pre-register the data you're going to use, the analyses you're going to do, and that reduces the degrees of freedom that you have with respect to playing around with your data. That doesn't mean that you can't do exploratory stuff as well, but for the stuff that's pre-registered, it gives you more confidence of whatever the finding is, which is often negative finding. And then there's lots of issues about ethics and data sharing and quality monitoring, et cetera. One resource, and again, these slides are up, I guess, on GitHub, so you can take a look. There are quite a few documents, if you go to this website here, which is the Caltech Institutional Review Board website, that you need to go to get approval for a study if it's human subjects work, but it also has lots of other resources. For example, it has data management and sharing document there amongst its 20 or so documents. And it gives you lots of links about what it is that funding agents, federal funding agents, well, all funding agencies pretty much, but these federal ones also, NSF and NIH, require in terms of data sharing. And if you go there, they're different. You have to specify what data types, what tools, what standards, and in general, if you're funded by NIH and NSF, but also for private foundations, and for that matter, if you publish papers, as you know, you're required, right, to share your data and typically also share your code. So you have to plan ahead and make sure that what it is that you share is in a format that's going to be usable for other people. So, Neuroscience Without Borders, or you have to use some other format, or BIDS for fMRI. You want to think about the particular types of data format and sharing. And then for humans, so for this particular study, for the Human Connectome Project, we only got access to a portion of it because, of course, one big issue there is privacy. And there's been quite a bit of work, and again, you would want to check. Depending on the details, it's quite possible to identify people, even if each of the variables available looks completely harmless. So we run into this quite often with the registrar. So they're human subject studies where somebody wants data from the registrar about Caltech students. So look, we just want anonymized data, no names, nothing else. And we're going to conclude a whole bunch of things from that about people's social behavior, decision-making, whatever it is. And you take a look, and well, they have the person's sex, race, age, and the house they live in. Well, for a lot of people, given Caltech's small size, I can narrow it down, and I can probably guess who you are. So even from very few variables, depending on the details, depending on the sample size, you can identify people. So there's lots of issues about privacy nowadays that, again, if you're analyzing an existing data set or sharing data, you need to think about. Lots of other questions. I'm not going to go through remotely all of them that we thought about before planning this study. So we look at the Human Connectome project. It has 1,000 subjects. That sounds like a lot. And we're going to ask, does brain activity predict something about personality? Well, it depends on how big your effect size is, right? 1,000 subjects may not be sufficient. And this paper, which is also on the background readings for this, has been often cited and made the point that depending on what it is that you're interested in, what level of confidence you want to have, you typically need surprisingly large sample sizes in order to have some confidence that there's an actually meaningful correlation between two variables. Hundreds, thousands for the typical kinds of effect sizes that we're looking at. But again, in a general study, in the ideal case, you would have some sense of what effect size am I looking at, and how many mice or humans, how many samples would I need in order to be able to detect a meaningful correlation with some confidence interval, right? And then, and you guys will go through this, in the remainder of the course, there are many, many different ways of analyzing your data, right? So for fMRI data, right off the bat, there's ways of cleaning it up and normalizing it and detrending it and regressing out various things. And you can keep going through all these boxes, and people have all of these different publications here, all these different colored paths, are different paths through this. Most of these are respectable and make sense. You can do different things. You can't do all of them. When you try a whole bunch, and people have gone through this exercise, you can get very different results depending on what analysis you do. And so there's no easy solution to, well, in principle, the solution is obvious, which is to do all of them and see how robust your results are with respect to the details of your analysis pipeline, right? Typically, you can't do that, and so you pick some subset that either spans a large range or pick some reasonable paths through this. But in general, you certainly want to do more than one. And definitely, you don't want to do a whole lot and only publish the one that worked, right? So it's obvious. And this is exactly what pre-registration would prevent. So just to finally put up the results from this paper, there were two data sets from each subject, resting state fMRI data sets, 1 and 2 on the X and Y axis. And this just plots how well we were able to predict, in this case, neuroticism, and on the right, openness, so these personality factors from resting state fMRI data. So this is the correlation of the one predicted by just a regularized regression versus the actual measured neuroticism, which we had available. And what you see is all these different colored dots here are different ways of parcellating the brain and different analysis pipelines. So we didn't try all of them. That would be a gigantic amount. But we tried a bunch. And what you would like is to see that they are kind of clustered together. And indeed they do for neuroticism, and in particular they cluster in this gray area here, which is not below the threshold we set for statistical significance of the findings from permutation distributions. And on the right, for openness to experience, they cluster sort of outside of that. So for this one, you would say there seems to be a weak ability to predict this particular personality dimension from resting state fMRI data. I can replicate it across two data sets, resting state 1 and resting state 2. The line of reproducibility, which is this diagonal between the two data sets, it's okay. I mean, they cluster around that, which is reassuring. but it depends on exactly how you analyze your data, and you can end up with non-significant findings or more significant findings, depending. So that's the kind of exercise you would want to go through. And of course, keeping in mind that these are small effect sizes and everything correlates with everything at some level. And so people have also published papers like this, which in general don't amount to any kind of explanation, but they raise the point that there are lots of confounding variables that may explain your data, which is one thing that you need to worry about. Just on this, for these kinds of variables, personality, intelligence actually is a good case in point. There are many variables to worry about. One of the ones to worry about here with respect to fMRI data is motion noise. So if neurotic people move more in the scanner than extrovert people, or if people with low intelligence, as is the case, move more than people with higher intelligence, depending on the range, you would pick that up in your data. And so that confounding variable, motion, that is correlated with intelligence or personality, would explain the relationship. So you would want to be very cautious in coming up with an actual explanation. For intelligence, we did exactly the same thing, same data set. A lot of these kinds of data, if they're sufficient available over some time, have the sobering picture like what's shown here. So the relationship probably that's been investigated the most with respect to some high order variable in humans and brain data is IQ, how IQ correlates with brain size, brain volume here, nothing functional. And over all of these studies, whose sample size is illustrated by the size of the circle here, you can see that the effect size on the y-axis, plotted versus time, the year in which the study was published, seems to get smaller and smaller and smaller. And that's a pretty common finding, that initially, that's certainly the case in statistical genetics, when particular polymorphisms in genes were associated with psychiatric disorders early on. Those were small sample sizes. They were published in Science and Nature. They were high impact. And they generally don't replicate. And over the course of time, with more and more studies and larger sample sizes, the general finding is effect sizes get smaller and smaller. This is a sobering finding for a lot of human research where psychological variables or psychiatric variables are linked to genetic or brain imaging variables, presumably because there are many other variables involved and many confounds. But so we looked at this, I'll just show you the final results here. Same representation here. We had the two data sets, 1 and 2, each I think about half an hour or so of resting state data from the Human Connectome project. And in this case, we're predicting intelligence from one of these little questionnaires like what you see on the internet, where you have like a bunch of pictures, Raven's progressive matrices, and you have to say what's the next one in the list, right? It's a sort of standard quick online IQ test. Same thing, though, that depending on how you analyze the data, you get somewhat different results. So you have to do a bunch, and if you really wanted to follow this up, of course, you could do this systematically, and you could say there, I can actually come up with an explanation that for a reason why this particular analysis pipeline does so much better than this one down here, right? But you would need to do a lot, and you would need to check. Here it just shows they're scattered, and it varies. It's still a very small effect, and so one point I wanted to make here is that I had this picture up before. One way of thinking about intelligence, which Spearman pioneered, was rather than using a single test, like what you typically get if you do it online, you estimate it across many different tests, which takes a lot more work. And so you can do that. I can give you the Wechsler Adult Intelligence Scale or something like that, which is across a whole bunch of different domains. So it would be memory and visual and verbal domains, etc. So it would take some time. And then I do some kind of a factor analysis, and I extract a single fluid intelligence factor, which Spearman dubbed G. So you could do that, and when you do that, and we did have that available in the Human Connectome project because these people had taken a lot of different tasks, what you find is that you can actually predict that measure of intelligence much better than the others. So this, which we had in the paper, this paper was an interesting, you might have thought that the reason we get these very weak effect sizes is because we're just limited by bold fMRI. And this is as good as you can get. Signal to noise is terrible. But it turns out we can actually do pretty well, and it's the behavioral measure of intelligence that was the problem, that if you use Raven's progressive matrices, this is not a very good estimate of intelligence. And so similarly, if you were looking at aggression or fear in a mouse, rather than using a single measure, you might have a whole battery of tasks, and you might look at, might extract a factor on the behavioral end to try to predict. Often we often think of trying to reduce the dimensionality of just the neural data, but it's equally important to think about the behavioral side, or for that matter, the sensory input side. Just to close on this, this applies to any large data set, but it's been particularly studied for these human fMRI data sets. And this is a paper that came out just recently, that it's very easy to manipulate these data to generate anything you like. So obviously, you can have adversarial manipulations. In that case, you're manipulating the testing data rather than the training data, and you're manipulating the testing data based on exactly what you know about the training data, such that your machine learning algorithm could just give you zero association. But in this particular paper, what they did was you can manipulate both the training and the testing data. This is a study looking at exactly what I said, which is it takes fMRI data, this functional connectivity matrix, so-called connectome, to try to predict IQ in people, big data set. And you can change it very slightly such that these enhanced data sets have a really, really high correlation with the original. So high that unless you knew what to look for, you would never guess that anything has been manipulated. But it has a large effect on the machine learning algorithm to be able to predict IQ. And it can increase it up to almost 1, even though it's very difficult to detect the difference. So it's easy to alter existing data. For adversarial manipulations, it's perhaps less of a concern because you're just going to get nothing. But for enhanced ones, you could set it up. I could release a data set that proves that we can predict intelligence almost perfectly from a large fMRI data set. And it would be very difficult to detect that that's been manipulated. That's the point that this paper makes. So with respect to the provenance of your data, if you're analyzing large existing data sets, but also if you're just getting data from any colleague or person in the lab that's sharing it with you, you want to know something about how the data were acquired, how it came about, and so on. All of these questions up here before you start digging into it. What's the time, Adi?
It's 10:20.
And we're going till 10:45? Okay. Okay, so that's sort of tells a story by way of one particular set of investigations that my lab has been involved in, that hopefully raises some general questions for all of you to think about. The provenance of the data, the trustworthiness of the data, and then just lots of checks, which I think there's no shortcut to just saying think about them very carefully. Think about the alternatives with respect to the sampling, with respect to the data analysis that you could have done, and ask yourself how robust are the results with respect to that. And definitely don't do what's the obvious thing not to do, which is keep playing around with data and with analyses until you get p less than 0.05 or something, right? Here's another example. So this one is purely psychological, actually. It doesn't involve any neural data, but pretty much all of the points apply to neural data as well, which is this one here. So this was started by my former graduate student, Chujun Lin, who's now about to start, or has just started as assistant professor at UCSD. And the question was, how do people infer whether something about a person from looking at their face, if otherwise they know nothing? So if you see a photograph of a face, you can judge that that person looks intelligent or trustworthy or extrovert, or make a whole bunch of judgments. And we do that all the time very quickly. How can we think about that? In particular, is there some low dimensionality to those kinds of judgments? So lots of other studies had done this before, and we wanted to do this in a way that we thought was better than the way that people had done it before, not knowing that we would run into lots of problems with reviewers. So we we had those. But we started, as we should, by pre-registering the study. So we pre-registered this. I think it was a series of pre-registrations. Since the reviewers raised lots of questions subsequently, we also did other stuff. So you don't necessarily have to publish only, in fact, you almost never publish only what you pre-register, but you would have the particular pre-registered component and then other analyses that were exploratory or that the reviewers forced you to do. We did what we have to do for all human subjects studies. So if you go to access.caltech.edu and you scroll down to the bottom, there's the, I don't actually know what it stands for, but anyway, CT is a set of online courses that you can take to get certified for human subjects research. If you're doing animal research, you would go through IACUC. If you're doing human research, you would go through IRB. But in all of these cases, you need to have approval by some board, the Institutional Review Board, or its equivalent, IACUC for animals, at Caltech. So we went there, we took the courses, we got certified, we put in IRB protocol, and then came up with this article here that started out in various other journals, including multiple reviews in Nature, before finally being published in Nature Communications. And along the way, running into lots of interesting questions and challenges from reviewers. But so let me just highlight a couple of points from this. The question is, how do people make judgments about faces? And so we wanted a bunch of faces as stimuli and a bunch of words on which people could rate them. And then just collect a lot of data, do dimensionality reduction, and see how those two were associated. So the first question is how to sample those. So for the face stimuli, we took existing databases that are out there. So there's a whole bunch that people had used. We used 3 of them. Those have a very large number of faces, more than people want to have time to rate. And so we projected these into a deep neural network that's better than humans at identifying, re-identifying faces. And we then sampled from a layer of this neural network using maximum variation sampling those 100 faces that are most representative of the total number of faces. So you have a large number of faces sourced from somewhere. So this limits, obviously, limits and potentially biases what you have as stimuli. But we took what we had, get a large number, and then try to ensure that we sample it in a way that's most representative, that spans the full diversity of all the different ways the faces can look, in this case, structurally to a neural network. So you could argue with different ways of sampling, but we did it that way. We split it up so it's 50-50 males, females, and those were the face stimuli. And then same thing for the words. We took 100 words from many established sources, and got like 1,000 words or something, whittled it down, and again, selected 100 that we thought were most representative. And in this case, we actually checked that these 100 words that we're asking people, so these would be labels like trustworthy, attractive, intelligent, whatever you could attribute to a person's face. We checked that these were representative in the sense that they corresponded, at least in this UMAP dimensionality reduction picture here, to what people would spontaneously generate. So we did a separate study. We just showed these 100 faces to people, asked them to come up with labels, and those are the gray circles here. These are just freely generated words that people spontaneously generate. Just give us a word that you think describes the person. And the ones that we used as stimuli, the red ones, were representative. So lots of work at the front end in just asking. We want to ask the question, how do people make judgments, which are a whole bunch of words, adjectives about people, from faces? How do we pick those adjectives? How do we pick the faces? And so sampling that took a lot of work. And then the finding was that there were 4 dimensions here from various dimensionality reduction techniques. We did a whole bunch, exploratory factor analysis, PCA, autoencoders, to check that this was robust. But they varied to some extent across different cultures. So we tested this. The participants that were actually looking at the faces and giving the ratings were across all of these different countries shown here. So we thought this was pretty good. We selected 100 faces on 100 words, much larger number of words than any previous study, carefully sampled across all these different 6 or 7 cultures in the world, and we found mostly 4 dimensions characterized. So that was the main finding. Reviewers had issues with this. And so here's one example from one reviewer that made the point, despite this praise, I have to state that the actual samples used are rather small. Despite all the excellent work, more data wouldn't hurt. Well, we couldn't collect more data at this point, but we could check whether or not the data that we had seemed like it was enough. And so this is doable. You just decimate the data and redo the analyses. And so we did that here. If you take out one subject at a time and you just do exactly the same analysis again, how similar are the results to the results that you had, right? And if things would start breaking down immediately, you would worry and you would think, well, I should probably collect more data. If it seems very stable until when you get fewer and fewer subjects, indeed, things will, of course, break down. You have some confidence that your sample size was sufficiently large. So we went through lots of exercises like this. And then we ran into the generalizability issue, and it was really reviewer 3 that raised these problems. But they raise a very important point and related to what I alluded to earlier. So he says, he or she writes, that we opened the abstract with a statement as a broad intro. People readily attribute many traits to faces. And this reviewer says, how can you say that? This sounds like it's all people and all faces and all traits. It's already way too broad. And you used English as a language here. And he or she goes on to say, English is just one of 6,000 languages. Why the limitation to English? Is this intended to be a study of English? And so I was inclined to write back to the journal and say, your journal has a problem. It's in English mostly. But no, this is a valid point, right? We had English as a language, and then also we had faces that were generally white neutral adult faces, what was available in these databases. So right away, for this kind of psychological study, you can see that the point is indeed well taken. To what extent would this generalize if it was a different language or if the faces were of a different race, of younger or older people, etc. And we had all those caveats in the paper, but the title did not make it explicit yet when the reviewers saw it. So this is a big problem. To what extent do you want to think about the way that the results that you have from the data would generalize? And it's something that's such a big problem that at least in psychology, but also for certainly for cognitive neuroscience and many other branches of neuroscience, I think it's a general, deep problem. And we did start thinking about this to design a follow-up study, but it's not obvious what the answer should be. So the question is, there's something in the human brain, or certainly in the human mind, where people reliably use these 4 factors to make judgments about faces. And so given the literature and given what you know, you can start thinking about this and you can say, okay, we used adult front-facing neutral white faces because those were available in databases mostly that we had. So you could say, okay, we're going to make it just racially more diverse and we're going to pick faces reflecting the demographics of America, right? But is that once you start thinking about that, that's not representative of what? That's not the right representativeness. You might want representative of the neighborhood in which the participants themselves lived, or not even where they lived, but perhaps where they grew up, since presumably that's where they acquired familiarity with these faces. Or if you think all of this is innate, you might want to say, no, the demographics of the environment way back when in which their ancestors lived. So you can see what the problem is. It's not obvious what representativeness, what a truly representative set of faces for this research study would actually be, right? And it's such a big problem, I put this in the reading. Tal Yarkoni had a paper in Brain Behavioral Sciences, aptly titled The Generalizability Crisis, where he thinks it's really big problem. It depends on your type of data. So for the type of human psychological data that I had here, it's probably a bigger problem than for some others. But to what extent do your results generalize to the point where you would actually feel happy that they answer the question that you're interested in? And that's not obvious. And in Tal's case, he has a section in his paper at the end where he recommends how to address this. And one of the recommendations is stop doing science and go into industry, which I wouldn't take seriously. You shouldn't do, but he thinks it's actually a very big problem. We also ran into problems just since this just came up a few days ago, with figures for this and for other papers. Some of the figures showed people, like faces, and we have other figures where they're not even people, but it's hands. And because of privacy and other issues, Nature Publishing said, no, you can't use those. So we thought, well, we can solve this. We just use DALL-E, and we generate, a generative artificial, a synthetic face or an image that looks very similar. No sooner had we done that than this came out, and it turns out Nature doesn't accept those anymore. So as it says down here, Nature will not publish imagery created wholly or partly using generative AI. And the reasons that they give here is the one that I mentioned, which is you don't know the provenance. You don't know what's behind how those were generated. And there could be all kinds of problems like the ones that are listed here. And so they've just decided not to use those. This may strike you as extreme, but it's there. We ran into it in actually trying to get figures published for this paper. It has, just at the very end, to conclude, you should not give up and you should not go into industry as Tagliarconi says you should because of all of these problems, one of which is the communication between this and my laptop. Let me try Okay. It has a happy ending because this paper won an award. Here's the authors. Chujun and Umit did all the real work. But despite all the problems, it ended up being a paper that addressed all of these broader issues, did a ton of work, and ended up in the title. You may have seen it back. It actually says English words. It has English in the title. So we thought a lot of what the reviewers harped on with respect to generalizability, checking sample size, and so on was too much at the time, but it ended up being a good exercise to do and actually won an award. So let me finish just in the last couple of minutes here. There's a lot more to being a responsible scientist and analyzing and certainly publishing data responsibly than what I've just said here. But here are just some general, obvious thoughts that obviously, but again, it bears repeating. You want to make sure that the data that you either are acquiring yourself or that you're getting from somewhere else have, best practices in research integrity, et cetera, connected to them. If you get data where you have any, suspicion that they have been manipulated, where there's any agenda or funding agency behind those data that are dubious, where you have questions, you need to check, right? So that's up to you, and it's a big space, but you need to know where they came from, and you need to check. And then, of course, there's a whole set of issues just related to general collegiality and, things, again, that should be obvious, but many of which are there in the Caltech Honor Code. That pertain to not stealing the data, right? Making clear that you have permission to use the data. If you are generating data yourself and sharing them, that you share them in a responsible way and share them so that other people can actually use them, et cetera, et cetera. We've had lab meetings on many of these issues. Co-authorship is often a big one. If you have data, it's typically not just yours, but it's generated in the context of a lab, collaborators, colleagues, and it's often a good idea before you start going down, doing anything with those data to ask everybody who's involved and even think about co-authorship if it's clear it's going to be heading towards a paper somewhere down the line. And then, of course, get approvals from IACUC or IRB if it involves vertebrate animals, which is what the IACUC is responsible for, or a particular vertebrate animal, humans, which is what the IRB is responsible for. And then just to finish here, you want to think about the social responsibility, the impact of the findings. And there's nothing, I listed a few things here, but there's no particular itemized list. I think you just need to think broadly, which is actually one of the reviewers for the paper that I was just talking about pointed out and went on about. He or she said, look, you can't publish this the way it is because this is what the news media may do with it. And here's some examples. And he was right. He or she was right. And so you want to think about how the data or any conclusions that you publish about them might be used or abused. And then be responsible and make sure that the data are shared, they're usable, the code is shared. Pretty much all publications now require this. I don't know about your experience, but in our experience, I would say it's sort of, I don't know, it's better now than it used to be, but it's often 50-50 in terms of how usable those actually are once you try and use them. So, and they require a lot of effort and curating to make sure that they are usable if they're data that are of any complexity. Okay, I want to finish with this very last slide, which is a theme that I started with because a lot of what I said in the last hour may seem a bit regulatory or dry to stimulate you for the rest of the course, which is that I think all of you are actually working on the mind. And you probably don't think so, but to first order, here are 3 ways of thinking about what the mind is. I think most philosophers and maybe also lay people think it's about conscious experience. I think that's a very problematic way of defining it. It may turn out that there's a close connection once people can decide on what they mean by consciousness, but that's definitely not the way to start. Certainly not if you're doing work in animals. There's a big literature on what people think the mind is, folk psychology and so on. You can put questionnaires out on Prolific and ask people, what do you think the mind is, or what do you think people's mental states are, et cetera. But that's just a question in social psychology, and that's not, I think, what most of you here are interested in either. You're interested in this, which is what should scientists think the mind is? And I think the answer is it's exactly what you guys are studying. If you're studying neural data of any complexity, the mind is whatever provides a causal explanation of behavior. So that's the causal link, and it's constituted by events in the brain, the kind of data that you guys are studying. So a lot of the analyses that you're going to be doing is analyses of neural data to put them into a format, to map them onto a model, to map them onto dimensions or factors that in some sense are actually psychological and would constitute what a scientific meaning of the mind should be that ultimately provides an explanation of behavior. So again, you might not think that you're doing this, but I think you guys are, and I hope that it actually provides you with some motivation to work on a discipline that's really poised to become a new discipline, and in the ideal case, replace what we mean by psychology with this new version of whatever it is, the data you guys are analyzing, which is neural, ultimately. How are we, or I guess I can look on time. We could take 5 minutes of, 5 minutes of quick questions or clarifications if you have them, and then we'll take a coffee break, and then we'll have Q&A for an hour after that.
Well, first of all, thank you for the talk. It's very pertinent to certainly my work. And one of the questions that I wanted to ask you is, you refer that big data sets, when they are analyzed by diverse approaches, the results depend a lot, they can depend on the approaches.
Yeah.
Can you please just elaborate a bit on this? Of course, a simple data set or a very low dimensional data set suffers less of this problem, but the bigger the data set, the more this will become. The more you use diverse approaches, the answers will not be easy to align.
Right.
Have you encountered this? What are your thoughts on this? And how have you dealt with that in your lab?
Well, it's a very good question, a very big question. Yes? Oh, sorry. The question is with respect to the different paths through a data analysis pipeline, all the different multiverse ways you can analyze the data, which typically consists of many different steps. How do we think about that? Does that become especially prevalent only for large sample data sets? Just, how do we think about this? I think it's relevant for any data set. And, well, one way of thinking about it is is to just simply ask how much flexibility or how many steps are there in any particular analysis. So if there's multiple steps, you need to remove outliers, you need to smooth the data, you need to normalize the data, you need to detrend the data, a bunch of things, and then you do some dimensionality reduction. there are decisions at all of those points. And to some extent, you can separate them. There's some that just have to do with removing what you think is noise, right? So there's like a whole bunch. And then immediately there's a decision, right? Well, what do you think is noise? And how stringent, where do you want to put that criterion? And again, I think the way to address it is to use a variable criterion. If I use really noisy data, less noisy data, super cleaned up data, and do exactly the same thing, how do the results look, right? And you would have confidence if they're really clean and slightly less clean, they look pretty much the same. And if they're noisier, they just get more and more variable and noisy. That would give you some confidence, right? If that doesn't happen and you get to a certain criterion and they suddenly totally change, then you might start to worry and you would dig deeper. And what you thought was noise maybe wasn't noise, right? With respect to the actual algorithms you use, or say you're using dimensionality reduction techniques of various kinds, right? So you could be using factor analysis, you could be using principal component analysis, you could be using independent component analysis, you could be using an autoencoder, you could have a whole bunch of different ones. And again, the question, once you start getting into the actual analyses often is what assumptions about the data or anything else does your analysis, does your model make, and are they appropriate? So for some, you might, for instance, think, well, this makes an assumption about linearity or normal distribution of the data, and those are violated, so that's not appropriate. For many analyses, like say factor, for all dimensionality reduction approaches, right? There are as many factors as there are data samples. And so the question is, how do you decide what the smallest number of factors is? Like that plot that I had up before, in the psychological study, right? So you can use exploratory factor analysis or PCA. You can get a scree plot. The more dimensions you add, the more variance you will account for in your data. How do you decide, right? And sometimes it's obvious, and there's just like some, very sharp elbow in your scree plot. It's clear that it's 4 or 5 dimensions that somehow work well and look different from the others. But often it's not. It's like very smooth, and you need to decide. So the bottom line is there are decisions all the way along. I'm not sure that it's necessarily different for very large data sets as opposed to small data sets. I think these issues apply everywhere. But I think, so the way to think about it is make a list, right? Or just jot it down of all the different steps, ideally everything that you would describe in a methods section of a paper sufficient for somebody to completely reproduce your results from the raw data, right? So what did you start out with? How did you sample the data? How did you exclude outliers? How did you do smoothing? How did you remove noise? Then what kinds of analyses did you do? Blah, blah, blah. Just spell it all out. And at each step, ask yourself, what could I have done otherwise? And what assumptions does this particular way of doing it make? But there's no shortcut. I think it's complicated, and it's at all stages of the analysis. But I think in the ideal case, before you do any analysis, you want to plan it and kind of make that list and say, here's what makes sense. Maybe ask colleagues if they agree. Yeah, Jeff?
I think one thing to add to that would be testable implications of your results, like for the Faces study, right?
Sure.
There's probably there's things that are not specific to this data set or to this method that are kind of scientific import that are predicted by these things, and you can kind of remove yourself from the food chain.
Right. Right. Yes. So Jeff makes the good point. Think about testable, further ways of testing your conclusions given your data. So some standard sanity checks would be, given the story I'm telling here, this should happen if I do something, and often it's something this should disappear, right? If I manipulated this variable, or if I took this thing out, or whatever. So you should play around. If it's clear that there would be such sanity checks, you should play around with your data and see that indeed it makes sense in that way. You would want to ideally test, so that you're going to test that it's robust with respect to different analysis pipelines. You would want to check that it replicates if you did it again. Now, so often these days you do, a sort of discovery study and a replication study, right? Or study 1, study 2, on entirely different samples of the same type of data. And again, you would need to think about generalizability. So you might want to just do a study in 2 mice, then the second mouse is a replication of the first. Or you have a whole batch of mice in one lab and a whole batch of mice in the second lab, and you do that study. Or you might want to say, I have these batches of mice, and now I have a couple of batches of rats as well, and I want to generalize over rodents, right, as opposed to just within mice. Depends how ambitious you want to be with respect to generalizability. Usually, we build in a little bit of that replication just within the data set, the way you analyze it, typically using cross-validation, right? So you would take some samples out, reserve some for testing, and that helps, but that's still internal to your data set. So in the ideal case, you would do all of these, right? You would use cross-validation within your sample and check that things seem fairly robust there, and you would have an entirely new data set thinking about the extent to which you want to generalize. Typically, you would not want to generalize, for example, across species, right? So you're not going to do a study in mice and say, let's see if it generalizes to people or flies, because you just think that it's not going to work and I wouldn't know how to do it. But you might want to have a different lab as collaborators, or just a different strain of mice, or whatever it is. Right? Yeah, I mean, these are all, so I guess this kind of segues into, so we'll break here, what we're going to do in 10, 15 minutes. But as you see, the questions, the topics are very open-ended, which is why maybe what people have done this, and maybe it's useful, and we could try to do it for this class, is to actually put up an exhaustive list of bullet points for all of this. I try to just boil it down by saying, just think and take your time, because there's too many, and it would depend on so many issues about your particular data set. But I think that, so bottom line, step back and think and ask yourself, why am I actually doing this? What's the question? And then how can I do this in a way that's most robust, generalizable, valid, and responsible? And there's no shortcut for just thinking about that. Okay, let's take a 10-minute coffee break, and then we'll go from there.